The other thing that eagles need is a quiet foraging area away from disturbance, and that's something we're more and more concerned about moving forward, is do we have the foraging space, sort of undisturbed foraging space for this population moving forward. Just a picture of some of the material that they have to use to forage with. Their talons are large, a couple of inches or so, so they have the material needed to hunt with. I want to show you just a little bit of information. This is a bullet camera that we put up in a bunch of nests. We had 18 nests wired back in 2002 and 2003 to look at the rate of both the identity and the rate of food coming into the nest, and then we were measuring chicks to look at growth rate related to provisioning rates. And interestingly, some of these nests that you see in the early period are just packed full of food. This is a nest up on the eastern shore of Maryland. You can see that there are 4 turtle shells here and a number of fish. And we oftentimes see that on the tapes, that these birds are stoking these chicks at a young age. As the nest gets later stage, you don't tend to see the available food laying around. And I think that's a strategy to really move growth rapidly in the early phases of development. But they have a varied diet, as you know from watching the cam. And this is from our study, almost 800 prey items that were identified. In the provisioning period, over 90% are fish. A small portion of birds, mammals. The mammals tend to be either muskrats or squirrels. We occasionally get a possum and other things in. I have also seen evidence of fawns being taken in some of the nests up on the Rappahannock. Some reptiles, and I'll mention that. One of the things that we've been interested in is turtle use, and I have been collecting turtle shells from under eagle nests for maybe 4 or 5 years now, and I'm interested in the distribution of species that they're using and that sort of thing, and I've maybe collected 300 or 400 shells. But they do use quite a few turtles. I'm not sure why. Because some of these turtles are about like that, and when you look at an eagle holding them, it looks like a little pill. So it's not like it's that much food, and so what the attraction is isn't completely clear to me. But they do seem to be attracted, and it varies between pairs. Some pairs you'll go to, there might be 10 or 12 shells under them, and then other ones none. So certain pairs seem to like certain things. Okay, so some of the requirements: they need adequate nesting substrate with good crown access, large habitat patch for nesting that's disturbance-free, close to water, and they need foraging habitat with adequate prey. I wanted to shift on now to a little bit about the history of our population and the annual survey. The bald eagles have been surveyed in the Chesapeake since 1956. They've been surveyed from the air since 1962. And we, Mitchell Bird and myself, Mitchell has been surveying since 1977. This is his 35th year doing the annual survey. It's my 20th. We've been flying together for a long time. The survey is really 2 flights. There's an initial searching flight where we fly the landscape, same landscape every year, up and down all the tributaries, to map new nests and to check on old nests. And then there's a second productivity flight. And so the productivity flight starts usually in late April. It goes through about mid-May, and we're checking all the nests that have been active to count the chicks in the nest and also to age them. And so we fly close down, right on top of the tree. We drop right in on the tree. We can look out the window and see the chicks. And so this is a little bit of the data that's been collected. And there are a couple things I'd like to point out to you. So this is the reproductive rate. This is the average number of chicks that are being produced per pair. What you'll notice here is that when the survey started in 1962, pairs in the bay were producing less than 0.6 chicks per nest. Okay, it requires 0.9 chicks per nest in order to replace adult mortality. So that's sort of the maintenance level. You have to have that in order to sustain an even population. We were way below maintenance, and that's the reason why the population was declining. We were not producing enough chicks to replace the adults that were dying every year, so the breeding population was in decline. DDT was banned in 1972. And we reached maintenance here, as you can see, about 1980, and we have never been below maintenance since then. So we have been producing more chicks than it requires to replace adult mortality, and so the population has been growing since then. One other thing I want you to notice is that we have one data point here in 1936. That's a survey that Brian Tyrrell did. It's the only other previous survey that was done on the bay. And so that is a pre-DDT estimate there. And you can see that we have now attained that. We are now producing chicks at a rate similar to what we think that the eagles were producing prior to the DDT era. Okay? In response to that, the population has been growing. It's actually been growing exponentially over this period here, with an average doubling time of less than 8 years. So every 8 years, the population has been doubling over this period, which doesn't mean a whole lot when you're down here and you only have about 30 or 40 pairs. But when you get to the point where we are now, in the early 2000s, we were at 400 pairs. All right, and now we're over 700 pairs. And so that's the thing with exponential growth. Once you get up to large numbers, a doubling means a lot. I mean, you can go from half saturation in terms of the landscape to full saturation in just a couple of years, right? And it's because of the numbers. The recovery goal was set here, and you can see this is the recovery goal was set for the Chesapeake Bay. We actually entered and exceeded in the mid-1990s. And now we are at about 4 times recovery goal. I want to show you what that looks like on the landscape. I'm going to show you a series of maps. These red dots here are nests for these different years. This was in 1970 when the population reached a low. I hope that you can see this. Let's go to the next one. It's in 1980. The next one is 1990. The next one should be 2000.
Wow. Oh, wow.
And then notice the difference between 2000, which was 10 years ago, and the 2010, which was last year. Okay. In 2010, we were approaching 700 pairs. I have not compiled the data from this year, but I believe that we're over 700 pairs. It's a lot of work to monitor the population of this size. I think that this year we have flown and looked at over 1,000 nest structures. And it's a lot. It's not like it was in 1990 when we had 100 pairs. Okay. So the population has grown from very low at the low, and we have had over a 34-fold increase over a relatively short period of time. Right? We're only talking about a few decades here. Okay. So we're now in a position to look back on sort of the Silent Spring era, which was the DDT era, and recognize the population change for what it was, and that is that the Silent Spring legacy was a relatively short-lived demographic perturbation, right? We reduced population, we reduced reproductive rates, they recovered fairly quickly after we banned DDT, and the population that has then affected a recovery. And so the effect of Silent Spring was relatively short-lived in this particular case. Really, I mean, we went from a low back to recovery in only about 2 decades. Okay. I like to say,
hungry.
I like to say a couple of other things about ecology, and then I'll say something about sort of where we're headed. And the first thing is, as we already discussed, eagles during the brood breeding period require aquatic prey, and there are implications of that in terms of their spatial distribution. This is a nest up on the Parappa, big blue catfish, which is sort of another story. Blue cats were introduced into the bay. They're not native, and much of the eagle recovery has been on the back of blue cats, which is an exotic, but I'll not go into that. So this is what we refer to as generally as the working area. It turns out that 95% of the eagle pairs that we have are within 3 kilometers of a major channel, and that's what this map shows right here, is what we refer to as the working area. This is area that is going to support the large population that we have. And it's because of this restriction. The other interesting thing that we have here in the Chesapeake Bay is a fairly steep salinity gradient. We have oceanic inputs here at about 33 parts per thousand. We have freshwater inputs at the fall line. And we have a steep gradient as these salt and no salt water come together. And it turns out that has important implications for eagle distribution. So these are the salinity gradients. Interestingly, the James has the steepest salinity gradient because it's closer to the mouth here. As you go up the bay, these different salinity zones sort of stretch out because they're farther and farther away from the source of oceanic inputs. And if you squint real hard, you might be able to see that the breeding density here in these higher saline waters is lower. The eagle nests are spaced out more compared to when you go up these tributaries into fresher and fresher water. Can you see that? Okay. If we look at that graphically, what this does is to break salinity zones up and look at density of breeding eagles as a function of salinity. And what you can see is that there's a functional break here at about 1 part per thousand salinity. So at below 1 part per thousand salinity, the population has been growing at a faster rate. This graph here ends in 2002. If we extend it out to present, the difference is about 8-fold. The density of breeding eagles in the lower salinity water is about 8 times higher than the other. Why is that? That was really the reason for Catherine's study. But the implication from a conservation perspective is that these low salinity waters here are supporting a lot of the chick production and a lot of the eagle pairs that we have in the state. So from a conservation perspective, in terms of effort per mile or importance per mile, these are the areas that we would focus on. It's the heart of the population. Yeah. One of the reasons that we believe that is true, that eagles are attracted to those areas, is because the fish densities are higher there. And this is from Catherine's video project. But there are 2 main fish that seem to be important to the population more so than the others. And those are shad, these anadromous fish that come into the bay to spawn, and catfish. And it turns out that shad and blueback herring spawn in waters that are less than 1 part per thousand salinity. So when they come into the bay, as you might know, American shad actually goes out into the ocean. It goes up into the Gulf of Maine and raises to reproductive age. It actually migrates down the coast and comes back into the bay to spawn. Where they spawn are these areas here that are in waters less than 1 part per thousand salinity, and we think that these spawning events are related to these higher density. The other thing that's true is that dabbling ducks tend to be concentrated in lower salinity waters. And so there just seems to be a lot of prey concentrated in lower saline waters. Yeah. Reese mentioned earlier the migration. The Chesapeake Bay is a convergence area for these populations along the entire Atlantic coast. They converge here in the winter from the north and in the summer from the south, in addition to ours. Well, it turns out that if you look at the distribution, let's go to the next one, of these migrant populations, they are in these areas. These are concentration areas that we've identified for migrants. Turns out they occur in less than 1 part per thousand salinity. So in terms of the conservation value of these nearshore areas, they're the most important for our breeders, and they're the most important for these migrants. And so they have great conservation value for birds along the entire Atlantic coast. So we have these different populations interacting, and in terms of our conservation responsibility, we need to be careful about the landscapes that occur within these high use areas. That's the general message. So low saline reaches support the core of our population. Highest breeding densities account for overall productivity. Low saline reaches also support the non-breeding, both our non-breeders, that is our juvenile birds, and these migrant population. And these concentration areas, these low saline waters, are somewhat unique. We don't have a corollary to that on the Pacific coast because we don't have a southwest population.